The Punnett Square is a diagram that is used to predict an outcome of a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach. The diagram is used by biologists to determine the probability of an offspring having a particular genotype. The Punnett Square is a tabular summary of possible combinations of maternal alleles with paternal alleles. These tables can be used to examine the genotypic outcome probabilities of the offspring of a single trait, or when crossing multiple traits from the parents. The Punnett Square is a visual representation of Mendelian inheritance. It is important to understand the terms heterozygous, homozygous, double heterozygote, dominant allele, and recessive allele when using the Punnett Square method. For multiple traits, using the fault line method is typically much easier than the Punnett Square. Phenotypes may be assessed using a Punnett Square, but the phenotype that may appear from a given genotype can be influenced by many other factors. Monohybrid cross. Mono means one. This cross indicates that we are examining a single trait being crossed. This could mean that we are looking at eye color. Each genetic locus is always represented by two letters. So in the case of eye color, say, B equals brown eyes, and B equals green eyes. In this example, both parents have the genotype BB. For the example of eye color we stated above, this would mean they both have brown eyes. They can produce gametes that contain either the B or the B allele. The probability of an individual offspring's having the genotype BB is 25%, BB is 50%, and BB is 25%. The ratio of the phenotypes is 3:1, typical for a monohybrid cross. When assessing phenotype from this, 3 of the offspring will exhibit the brown eyes and only 1 offspring will exhibit the green eyes. The way in which the B and B alleles interact with each other to affect the appearance of the offspring depends on how the gene products interact. This can be things like lethal effects and epistasis. Dihybrid cross. More complicated crosses can be made by looking at 2 or more genes. The Punnett Square works, however, only if the genes are independent of each other, which means that having a particular allele of gene A does not alter the probability of possessing an allele of gene B. This is equivalent to stating that the genes are not linked, so that the two genes do not tend to sort together during meiosis. The following example illustrates a dihybrid cross between two double heterozygote pea plants. R represents the dominant allele for shape, while R represents the recessive allele. A represents the dominant allele for color, while A represents the recessive allele. If each plant has the genotype RAA, and since the alleles for shape and color genes are independent, then they can produce 4 types of gametes with all possible combinations. Ra, 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 and Ra. Since dominant traits mask recessive traits, there are 9 combinations that have the phenotype round yellow, 3 that are round green, 3 that are wrinkled yellow, and 1 that is wrinkled green. The ratio 9:3:3:1 is the expected outcome when crossing 2 double heterozygous parents with unlinked genes. Any other ratio indicates that something else has occurred. Fault line method. The fault line method can also solve dihybrid and multihybrid crosses. A problem is converted to a series of monohybrid crosses, and the results are combined in a tree. However, a tree produces the same result as a Punnett Square in less time and with more clarity. The example below assesses another double heterozygote cross using RYYXRYY. As stated above, the phenotypic ratio is expected to be 9:3:3:1 if crossing unlinked genes from 2 double heterozygotes. The genotypic ratio was obtained in the diagram below. This diagram will have more branches than if only analyzing for phenotypic ratio.